Okay, I think I am audible. Shuna paisa no? Okay, let's start the class then. So we were discussing sorry, we were discussing the chapter photosynthesis, and I think we have completed the first class. In the first class, what we have discussed was first class or second class? Second class we have gone. Today is the third class, right? Okay, so what we have discussed up till now is we have talked about the general points about photosynthesis, right? As far as what is photosynthesis, how is it depicted in the form of an equation, whether it's an oxidation reaction or it is an anabolic or a catabolic reaction. This is the fact, these are the informations that we have already discussed. In the last class, I think we were discussing something called as requirements for photosynthesis, and whenever we talk about requirements, we talk about 5 types of requirements in a broader sense. We have talked about carbon dioxide, we have talked about water, and I think we have talked about light also. These 3 we have completed, and we have started with the next one, that is pigments. I think we have completed the chloroplast also. Out of the 5, we are discussing the 5th one, that is pigments. Whenever we talk about photosynthetic pigments, we understand that whatever might be the organism, any organism that is conducting photosynthesis, no? Either it's a eukaryote or it's a prokaryote, they will have their own photosynthetic pigments. Talking about pigments, we remember that pigments has been broadly classified into 3 categories, that is, ki asile pigments bilak? Photosynthetic pigments ki asile? Chlorophyll, carotenoids and phycobilins, right? In case of lower organisms, we have talked about the word called as phycobilins. Last class of ami chlorophyll or discussion korisu, we have talked about the structure of chlorophyll. We have said something called as the universal chlorophyll, etc., etc. So today we will be starting with the next photosynthetic pigments, that is carotenoids. Heading koriba carotenoids. So what we will do is we will write the note first and then we will explain. Likhi jua first. So if you are talking about the first point, we are talking about the color of carotenoids. Photosynthetic pigment we have discussed about chlorophyll. Our chlorophyll discussed kore ki koise, these are nothing but green colored pigments, right? And when we talk about carotenoids, generally yellow, brown, red, orange, etc. Etiolated, E-T-I-O-L-A-T-E-D, etiolated plants have the color of carotenoids. Okay, the next photosynthetic pigments is carotenoids. Obviously, it's responsible for the coloring, right? Apart from green, baki colors kihor karne hoy carotenoids. Light is not essential for its synthesis. Why we are writing this point? Because we are making a comparison between chlorophyll and carotenoids. If you remember, I think we have discussed something called as a precursor of chlorophyll. Biosynthesis of chlorophyll is something that we have done in the last class, and we know who forms chlorophyll. If you remember, chlorophyll kune form kore? Last class আমি কৰিছিলো. Who forms chlorophyll? If you have forgotten, just turn your pages. You will see. Refer to this section, biosynthesis of chlorophyll. Can you see something called as glycine? Right? That part. Ultimately, last of ki ase? Chlorophyll ase. Chlorophyll kune form korise? Saw something called as protochlorophyll. Yes, and protochlorophyll, chlorophyll majot ki likha ase? Light. Right? So, what we have explained, until and unless light is available, chlorophyll will not get formed, right? Protochlorophyll, in order to make chlorophyll, ki lagibo? Light lagibo. So, that is a pigment which is light dependent, but in case of carotenoids, light is not essential for its synthesis. Light hulehe je carotenoids hobo teneko eko kotha nai. Okay? Next, as far as the function is concerned, this is a word that you need to remember. These are called as what? Protective pigments. Whom do they protect? They protect the chlorophyll molecule. I think you all remember we have talked about something about the pigments in our introductory class. What we said is whenever we are talking about photosynthetic pigments, these pigments will always remain in a group at not single. I think you remember we have discussed that, right? Pigments will like in the form of a group, and when we are talking about a group, it is not only chlorophyll that is present only in the group, it is the accessory pigments, the carotenoids are also present in the group, right? So, we have a group, in the group we have the essential pigment, we have the accessory pigment also. Who forms the essential pigment? Obviously the chlorophyll, and the accessory pigments is the carotenoids. So, hey group 2, so what's the important kunto? Obviously chlorophyll. That chlorophyll molecule, if it comes directly in contact with the sunlight, we see a process called as photooxidation. Chlorophyll molecule to bhangi jabo, right? Basic sunlight he trap koribo nware. So, direct sunlight ketiao chlorophyll a molecule nopore, right? No doubt we say photosynthesis starts as soon as light falls in the pigments, right? Pigments will take in the light and the process will start. Talking about pigments, we know chlorophyll, carotenoids all stay together, but which pigments take the light first? It is always the accessory pigments that take the light first, and then they transfer it to the essential pigment, that is the chlorophyll pigment. Why? Because if chlorophyll pigment comes directly in contact with light, what will happen? Oxidation, photooxidation, oxidation in presence of light. In a very simple language, what do you understand that? Direct ahile chlorophyll molecule to bhangi jabo. If the chlorophyll itself gets broken down, photosynthesis to eneo no hobo, right? Okay. So, who protects? It's the carotenoids. So, what is the function? Protective pigments. Next is they act as accessory pigments. We understood just the opposite of essential is accessory. Also present in chromoplasts. We are talking about plastids in case of flowers and fruits, and they are responsible for giving the color of the fruit and the flowers, right? Flowers bilakor petals bilak colorful hoy, fruits bilak colorful hoy, because of what? Obviously pigments. And where are the pigments present? In the plastids. Which plastid, to be precise? We are talking about something called as chromoplast. Next is etiolated plants have the color of carotenoids. I think we have discussed about this etiolation process to discuss korisilu monte ase. Remember, we have talked about etiolation. Okay, anyway, forget. So, what is etiolated plant? A plant that undergoes etiolation is called as a etiolated plant. Now, what is etiolation? Okay, let's understand with the help of a example. We are taking two seeds. Okay, so A seed 2, this is seed A, this is seed B. This seed is exposed to sunlight, and this seed is not exposed to sunlight. Sunlight 9. Since we are talking about a seed, obviously the seed will germinate, right? Even in this condition, no doubt light is not there, seed to germinate koribo nokore. Light থাকিলেহে germinate koribo, tenikoa kiba kotha ase niki? Light noholeo to germinate koribo. We have the seed, and from the seed obviously the baby plant will come out, right? It will happen in presence of light, it will happen in absence of light also. But if you are comparing this plant and this plant, they will have certain morphological differences, right? What is the difference that you will see? Adult plant jonto niki sun ba lightor tolot dhuniyake germinate korise aru adult jonto shade out hoise, thik ase hei duita plantor majot okoman holeo difference dekha pam ne nabo. Especially the difference in terms of what? We are not going into anatomy, we are not going into very deep knowledge. The basic difference that we see is something in color, right? Eto ki hobo? Sunlight ase dhuniya ke chlorophyll form koribo. Chlorophyll kyo form kore? We have already discussed the chlorophyll form koribo lagile ki lagibo? Light lagibo, right? Jedi light nai, chlorophyll form koribo jano? No kore. So what will happen to the color of this plant? They will become what? Something whitish or pale in color. So this condition is called as etiolation, right? So etiolation ki, if a angiospermic seed is exposed to no light condition starting from the point of germination, the symptoms that we see in a plant is called as etiolation. And etiolation thaka plant to kami ki boli kau? Etiolated plant. So etiolated plant will not have chlorophyll, green na thakibo, no? Ki thakibo? Different colors, who is responsible? Carotenoids. Is the point clear? Right? So starting from germination, jedi plant dal hoy, tar jedi light na thake, tar color bilak fade hobo, fade green color no hoy. Kyo no hoy? Karon light nai. Right? So and this plant is called as the etiolated plant. Next is just, this was about carotenoids, and just an extra point, something about the deciduous forest. Deciduous plants, janu ne? Evergreen plants and deciduous plants. We can classify plants under two categories broadly, no? Evergreen mane ki? What is the basic difference? Okay, come to deciduous or evergreen. Evergreen is something that they will never shed their leaves. And if you are talking about deciduous, every season, by every particular time, they will shed their leaves. So we are talking about the deciduous forests. Deciduous forests are called as colorful forests. They are also called as autumn forests. During the autumn season, especially those trees who shed their leaves, before shedding their leaves, hey, leaves bilak colorful hoi jay na jay. Before they remain green, and after that, as the autumn season comes or arrives, what happens? Their color changes, often yellowish, orangish hoi jay. At that time, we see the color of what? Carotenoids, right? So deciduous plants ki kore? Shed their leaves. Shedding or homoy jintu color thake, that color is because of ki carotenoids. Is the point clear? Right? Okay. Now we have to talk about classification of carotenoids. Next point likha, carotenoids are of two types. Yeah. Likha, carotenoids are of two types, carotenes and xanthophyll. So we'll write certain points about carotenes first. Right. So carotenes or key information janiba, the basic carotene question is asked about the basic carotene. Basic carotene is called as lycopene, and lycopene is present in colorful bostu bilagot, no, especially tomato, chili, etc. Now there are three main isomers of carotene, three types of carotenes, alpha, beta and gamma. Out of that, what is important is beta carotene, which gets converted to vitamin A, and that is the reason why we are asked to eat colorful vegetables, colorful fruits. Khabol diye no kiho karne. It's not like this that we eat a tomato and then we become red. Teneka to nohoy no, karon pigment to kot jabo. Ba kiba eta bostu jodi green color chili eta khala ba yellow chili eta khala, we don't become yellow. What happens to the pigment? It gets converted, right? It gets converted to what? Vitamin A. So this is the point that is important, the first point that is important. That's all. Carotene formula to likha sir. Kuntu? Beta carotene gets converted to vitamin A inside the body of the animals, by in animals, right? So anything with carotene to be jodi khua, tetia hole ki hobo, inside our body it will get converted to vitamin. Oxygen derivative of carotene. Right? Xanthophylls basi iman important nohoy, khali example kita we can just remember. As it is, it's easy. Xanthin word kita ase, so it's easy to identify, right? So what is xanthophyll? It is a photosynthetic pigment. It's almost equal to carotenoid, kintu carotenoid or ki ase carbon hydrogen ase, yate ki thakibo carbon hydrogen aru oxygen thakibo, right. So, we are writing it oxygen derivative of carotene, right, that is all. So, we have talked about the two types, that is chlorophyll and carotenoids. The next type of photosynthetic pigments is phycobilenes. Heading koriba phycobilenes. Phycobilenes as a pigment ক'ত দেখা পাম কোনবিলাক organisms কোনবিলাক photosynthetic organisms. Remember a word called as phycoerythrin, phycocyanin. মনত আছে এইখিনি আমি ক'ত পঢ়িছিলো again phycoerythrin two types আমি পঢ়িছো R phycoerythrin আৰু C phycoerythrin. We were talking about red algae and BGA, right. মই পিছৰ পাৰ্টটো শুনা নাই তোমাৰ কোৱেশ্চনটো ফাইকোসাইনিন কি হয় কি লিখা আছিলে actually your voice is cracking মই একেবাৰে ধৰিব পৰা নাই তোমাৰ পিছৰ পাৰ্টটো কি কৈছা I heard phycocyanin later part I could not hear oh color blue okay Allophycocyanin ALLOPHYCO cyanin CYANIN okay এইটো তোমালোকৰ point of confusion লাগিব নালাগে no need to write this one. Talking about the structure, actually the question has never been asked as to what is the structure of carotenoids, right? But we need to know ketiyaba ahi jabo pare because we have already studied something called as closed tetrapyrrole structure in the last class, right? Closed tetrapyrrole structure kar asile? What we understand by closed tetrapyrrole structure, something like this. Remember we have drawn something like this, right? Mg mazot asile, yate saita ring asile, pyrrole ring, kar structure rakhiso? Chlorophyll. Right. So, chlorophyll or ki asile closed, yate ki ase open. We will see the pyrrole rings arranged in a linear form, right. Yate saita pyrrole ring eneke ase. In this case, the pyrrole rings would be arranged like this, right. Iman details na lage. This question is not asked, but chlorophyll or question to ahe. So, this is all about the requirements. So, up till here we have discussed the 5 different types of requirement in order to conduct photosynthesis, right. After this, we have to talk something about the pigments. Ekhini hol? Sobore hol? Can I rub the board? Online students? Last row gita? Thik ase likhibo. For those who have finished, heading kora arrangement of photosynthetic pigments. Is it over? Okay. So, during the process of photosynthesis, we know that light is taken in by the plants, and whenever we talk about taking in of light, who is responsible? Obviously the pigments. Some of the discussion we have already done talking about pigments or talking about the arrangement of pigments. What we have discussed as far as where will you find the pigments? Obviously inside the chloroplast, and to be precise, kot pam? Thylakoids pam. আৰু তাতকেও যদি ডিটেইলছত যোৱা outer membrane of thylakoid, আৰু তাতকেও যদি ডিটেইলছত যোৱা it is in the lipid layer of thylakoid. I think we have written all this information before. That was the first point we know. Pigments ক'ত আছে? Thylakoid. Second is, as far as the arrangement is concerned, we know they are never single, they are arranged in a group. Etu kothaও patisu. Today we are going to write something about that. Whenever we talk about a group of pigments, no, photosynthesis into group associated, we have a word that is called as a quantosome. So, হেতুৱে লিখিম এতিয়া লিখাচোন Number of pigment, yeah, this point, second point. Okay, number of pigments or number of pigment molecules required to produce the photochemical reaction. So, which photochemical reaction we are talking about? Obviously, photosynthesis. So, photosynthesis arambho koribo lagile, the number of pigments that are responsible, that group is called as what? Quantosome. So, what is a quantosome? It is nothing but a collection of pigments, right? The first group that is responsible for the beginning of photosynthesis, and I think you remember a common brief idea to already disilu, no? Photosynthesis keneke hoy. What is the first step of photosynthesis? If you remember, we have talked about something called as release of an electron. Koisilu last class on. So, hey, release of an electron is considered as the first step of photosynthesis. Hey, electron to release koribole lagile, junto group of pigments associated hoy, hey group to kami ki buli kom? Quantosome, right. So, what is the question that is asked is photosynthetic unit is also called as dash, or what is a quantosome? Quantosome is nothing but a photosynthetic unit. We use the word unit whenever we need to measure something. What we are measuring? The number of molecules that is present in a group, right. Pigmentum thakibo group, obviously chlorophyll basically thakibo, and tar upori ki thakibo? Accessory. Whenever we talk about accessory, we are referring to what? Carotenes and xanthophylls, right. So, remember this word. And if you have read the chapter, you have come across a word called as a reaction center. Jedi porhisa no photosynthesis to jedi agote porhisa. Reaction center ami discuss korim. Tamane swa eta group of molecules thakibo, right? Ami draw korim olop pisor etia na lage. So ei gutei group to ami ki boli koisu? Quantosome. Yate ki thakibo? Chlorophyll aru xanthophylls. Out of this, you will always have one member that is very, very essential. Obviously, whenever we talk কি হবো গুটে গিটা পিগমেন্টসৰ ভিতৰ সবচে এসেনশিয়াল পিগমেন্ট কি হবো ক্লোরোফিল হবো and if you are talking about chlorophyll we have already discussed around 9 types of chlorophyll and out of the 9 types সবচে essential কোনটো বা universal কোনটো chlorophyll A. So in this group they will have a reaction center, and what is the reaction center? Nothing but chlorophyll A molecule. কিমান chlorophyll থাকিব? 230 chlorophyll থাকিব, কিন্তু chlorophyll A only one. And that is that cell from where, or that is the molecule from where the electron will move out, right. So, the most important member of this group is called as a reaction center. We will be discussing, all of us are discussed for years. Is this part clear? Quantosome clear? Can I rub the board? Oh. Since we are writing it in the form of a note, we'll ekdam serially ami bhalke likhi jam. In a proper sequence. Eta before going further, ketaman points likhi lo. Eto to janu sake. If you notice, you don't have to write, okay? Or you can write it down also, doesn't matter. Write it down. এইটোতো আইডিয়া আছে আমাৰ actually photosynthesis হয় কি two steps light reaction dark reaction light reaction কি হয় cyclic photophosphorylation and non cyclic photophosphorylation who is responsible group of pigments dark reaction under the heading C3 cycle C4 cycle and CAM cycle this is the way we are going to study this chapter okay so points of first point likhiba So photochemical, thermochemical, we understood. Next, who discovered dark reaction? It is Blackman who discovered dark reaction. It is one question ahe. Blackman has studied something called as the Q10 factor, and then he has discovered dark reaction. Talking about dark reaction, as you all know, we'll be talking under the heading C3, C4 and CAM cycle. C3 cycle is also called as Kelvin cycle. Why is it called as Kelvin cycle? Because he was the one who has given the cyclic pathway of the dark reaction. Discovered dark reaction are giving a cyclic pathway to a dark reaction duitai beleg beleg. Discover kune korise Blackman. What does Kelvin do? Gave the reactions, right? Ki bostu ki hole convert hoy no cyclic eta process C3 cycle buli kau no gute reactions bila kune dise Kelvin aru he karone tak ami ki buli kau Kelvin cycle buli kau. Now talking about Benson, he has studied something called as Q10 factor. Q10 is something called as temperature coefficient. এটা ৰিয়াকশ্যনৰ টেম্পাৰেচাৰ কোফিচিয়েণ্টৰ ওপৰত বেচিছ কৰি পেলাই ডাৰ্ক ৰিয়াকশ্যন দিছিলে what is Q10 ডিটেইলছ নালাগে right maximum question কি আহে যে what is the Q10 for light reaction what is the Q10 for dark reaction we have two values assigned for that how did the value come or how was it deducted he process to among nalage kintu Q10 just jehetu word to ase okoman janibo lagibo what is Q10 just understand suppose we are talking about a reaction where A is getting converted to B maybe around 40 degree Celsius. Suppose, right? Eta reaction ase A kihole convert hoise B le convert hoise temperature kiman lagise 40 degree. If you want to double the rate of reaction, what he has said that if you want to double any form of a reaction, what you can do is increase the temperature by 10 degrees Celsius. That is what is called as Q10. So, what is Q10? Increase in the temperature. Why do you increase? To double the rate of reaction. How much do you increase? Just a rise of 10 degree. Etu suppose A to B convert hoise dhora 2 times kiba eta produce korise, thik ase 4 times jodi produce koribo lage A to B yate to already 40 degree Celsius asile plus ami ki korim 10 degree Celsius hoy gole eta 2 times to 4 times ho jabo. Ekdom simple ke, you don't have to write sentences, you don't have to by heart, nothing. What is Q10? A Q10 is a factor that is associated with what? Increase in the temperature of a reaction. What will happen if you increase the temperature of a reaction? The rate of the reaction will become doubled and faster, right? What is the amount that you will increase? It is by 10 degree, and so we have the term called as Q10. Q, temperature coefficient, how much will you increase? 10 degrees Celsius, that is all. Bujhi paisa? In whatever reaction it might be, right? And it is not like this, that if you want to increase something or if you want to fasten a reaction, it is not only the temperature that is essential or that is important to be increased. With the temperature, yate jibilak chemicals asile no, kisuman to chemicals associated hoy asile, so chemicals bilako increase koribo lagibo. Suppose yate dhora X chemical use kori asile, yate ami kiman use koribo lagibo, maybe twice X, maybe 4X. So it is not only temperature, but all the factors you need to double, then only the reaction will be doubling or it will be faster. But since we are talking it from temperature point of view, what we are saying is if you want to fasten or if you want to double the rate of a reaction, you have to increase the optimum temperature or the required temperature by 10 degree Celsius, right? Moi likhai dim bostu to Q10 bostu to buji paisa. Reaction eta double koribo lagile tar jiman temperature hoy asile agor temperature tutke 10 degree borhai dibo lagibo. That is what is called as Q10. It is called as such because that food to prepare hoy no, temperature associated food. What is Q10? It is a temperature coefficient. What we understand, rising the temperature by 10 degrees Celsius in a reaction, that will double the rate of a reaction. 10 degree ketia diba in the optimum range. Did you have you heard the words called as minimum, maximum and optimum? Janune eta reaction or tinta word ase. তিনটা cardinal points আছে, we will be discussing cardinal points later, but as of now এটা reaction যেতিয়া আৰম্ভ হয় ন এটা টাইমত maximum হয় আৰু তাৰপিছত gradually কমি যায়, right? So similarly we will have 3 points relating any type of a reaction. The words that we use is minimum, maximum and optimum. Minimum কি হয়, it is that point where the reaction has just started. Optimum কি হয়, the reaction is at its best in that point, and what is maximum, beyond that point the reaction will stop. That is called as maximum. So আমি যে temperature to increase কৰিছো কেতিয়া increase কৰিম, যেতিয়া reaction to optimum range ত থাকিব, means best ত থাকিব. Understood? And sometimes the question is asked about these values, no? কেতিয়াবা, not very important, don't by heart this statement, একো নাহে, কেতিয়াবা আহিব পাৰে যে light reaction I mean tomar Q10 value key, not very important also, okay? Okay, so this was the third point. Intermittent, M-I-T-T-E-N-T, no? Intermittent. Was Warburg, W-A-R-B-U-R-G. Warburg has carried the experiment in a green algae that is called as chlorella by using intermittent light. ব্ল্যাকমেনে ডিস্কভাৰ হে কৰিছিলে হয় নহয়, সেই এক্সপেৰিমেণ্টলি প্ৰুফ কোনে কৰিছিলে বা সেই তেনেকুৱা বস্তু এটা যে এক্সিষ্ট কৰে সেই বস্তুটো কোনে প্ৰুফ কৰি দিছিলে, এক্সপেৰিমেণ্টটো ৱাৰবাৰ্গে কৰিছিলে, ৰাইট? ব্ল্যাকমেন said that there is something called as a dark reaction. He has also done his own experiments. Blackman experiment কৰিছে কিন্তু কনফাৰ্ম কোনে কৰিছে, ৱাৰবাৰ্গে কৰিছে, that Blackman is correct. ৱাৰবাৰ্গে কি এক্সপেৰিমেণ্ট হিচাপে লৈছিলে, chlorella. Chlorella we know na green algae, and that ki lagisil obviously light. Now we have something called as intermittent light. What is intermittent light? Light given at intervals. Yate aru eta point janibo lage. Suppose we are taking two plants, okay? A2 plants out something called as okay, A2 joar agote light could be divided into two types, continuous aru intermittent, right? কণ্টিনিউয়াছ লাইট হ'ব পাৰে, ইণ্টাৰমিটেণ্ট লাইট হ'ব পাৰে। তাৰমানে কণ্টিনিউয়াছলি এডাল প্লান্ট তুমি ডাইৰেক্টলি লাইটৰ আগেইনষ্টত এক্সপ'জ কৰি থৈ দিছা, অৰ এ প্লান্ট কুড বি এক্সপ'জ টু ইণ্টাৰমিটেণ্ট মিনছ what? এবাৰ লাইট, এবাৰ ডাৰ্ক, এবাৰ লাইট, এবাৰ ডাৰ্ক। If that is the condition, that is called as intermittent. So we have two plants. In this, if suppose this plant is exposed to continuous light and this plant is exposed to intermittent light, right? So এইটো প্লান্টৰ লাইট পাইছে, এইটো প্লান্টেও লাইট পাইছে, তো ইয়াতো কি হ'ব? ফটোসিনথেসিস হ'ব, ইয়াতো কি হ'ব? ফটোসিনথেসিস হ'ব। Now you have to compare as far as the rate of photosynthesis কাৰ higher হ'ব? Intermittent lightত rate of photosynthesis higher হ'ব নে continuous lightত? কারণ we know if photosynthesis has to occur, obviously the plant will require light. Is it continuous light that it require or it intermittent light? It is always intermittent. Why? Continuous light dile ki hobo? Continuous light dile okol light reaction hobo. Light reaction hole faster the light reaction, faster would be the dark reaction. Dark reaction tor karone ki produce hobo? Food. Iman food produce hobo যে it will start accumulating in the cells and ultimately it will make the process slower. কিন্তু intermittent হলে কি হ'ব? Intermittent light reaction complete হৈছে, dark হৈছে, darker বস্তুখিনি use হৈছে, আকৌ light reaction হৈছে. So you produce something, you use it off, then you produce again. That makes the reaction faster. যিকোনো এটা বস্তু যদি accumulate হৈ যায়, accumulate হলে হলে processটো কি কৰি দিব? Slow কৰি দিব. So what you need to remember, photosynthesis is higher, continuous light or photosynthesis, ক'ৰবাত লিখি থৈ দিয়া, photosynthesis, sorry, slower, or intermittent light or photosynthesis higher, right? The rate of photosynthesis is always higher or highest during intermittent light, when the plant is exposed to intermittent light as compared to continuous light. So these were the points. So we were discussing about photosynthetic pigments. We have talked about the different pigments. We have talked about its arrangement also. Next, we have to say as something called as photosystem. Light reaction or key involved হৈ আছে? PS1 আৰু PS2 অলপ আগতে চাৰ্ট দিছিলোঁ ন, photosynthesis কি হয়? Light reaction, dark reaction. Dark reaction of C3, C4, C3, C4, CAM, right? Light reaction of মই কি দিছিলোঁ? Something called as PS1 and PS2. So we'll be discussing about this two now. As far as the word PS1 and PS2 is concerned, what we know? Photosystem 1 and photosystem 2. By the word photosystem, what we understand? Actually, PS বুলি ক'লে বা PS1 বুলি ক'লে কি হয় বস্তুটো? What is PS1 and what is PS2? PS1 to বাদে দিয়া. What is PS? Photosystem. Photosystem মানে? Light reaction or দুটা হয়, দুটা বস্তু আছে, PS1 আৰু PS2. Actually, PS1, PS2 কি হয়? Instead of by hearting, no, we should know also যে PS1, PS2 কি হয়? This is nothing but হয় নহয়? Right? So PS1, PS2 কি হয়? দুটা গ্ৰুপ. কিহৰ গ্ৰুপ? পিগমেন্টছৰ গ্ৰুপ। যেহেতু লাইট ৰিয়াকশ্যন, লাইট ল'ব, কোনে ল'ব? পিগমেন্টছে ল'ব। পিগমেন্টছবিলাক কেনেকৈ থাকে? গ্ৰুপছত থাকে। কিমানটা গ্ৰুপ এছ'চিয়েটেড? টু গ্ৰুপছ। হোৱাট আৰ দ্য নেম অফ দ্য গ্ৰুপ? PS1 and PS2. Clear? So what is PS1 and PS2? These are nothing but groups of pigments. Next heading kora, two pigment system. Next is we'll draw a chart where we will compare the two groups. So, ekhon chart aka. So, PS1 and PS2 ki hoy nothing but two groups that is responsible for the light reaction. So, we have to compare the two groups, and I think you all remember whenever we talk about it as a group, we know what the group comprise of. It comprises of pigments, and which pigments? Chlorophyll and accessory pigments, right? And I have already said that whenever we are talking about a group work, hey group ba team eta form korile hoday eta important member thake ba leader thake, right? So, similarly, if this is a group and that is a group, they will also have an important member or the leader, which is called as key reaction center. Clear? So, we will come to this word first. Reaction center means the most important molecule of this group would be P700 and P680. P mane photosynthetic, right? 700 and 800 talks about what? It talks about the wavelength of light it absorbs. Right? We have talked about something called as a light spectrum, no? Koisilu no? Absorption spectrum and eta asile absorption, eta hol action, right? And what we know, rate of photosynthesis kuntu light or besi hobo? Red light, as compared, rate of photosynthesis will always be higher on the redder side, right? It's always between 690 onwards, right? So, what we have is red light or jehetu photosynthesis besi hoy, so we are taking 700 and 680. This falls under which range? Red light itself, right? Ami P400 to likha nai, P600 to likha nai. Remember something called as PAR, photosynthetically active radiation, the range of light that plants take in to do photosynthesis. Light puli kole to bahut wavelengths ase no, kunkhini wavelength plante loy 400 or 700, ekini wavelength ok ami ki boli koisilo PAR, because the plants take this. Apart from photosynthetic bacteria, baki sobe maximum of the plants e ki loy PAR, right? We have talked about UV light, we have talked about visible light, and we have talked about infrared light, right? As a visible light or junkhini wavelength hoy, e gutei khini ami ki boli kau PAR boli kau. And among this wavelength 400 to 700, e gutei khini bhitorot highest photosynthesis kot hobo red light, right? So, we are talking about P700 and P680. Question hoday khude, what is the reaction center of PSO1 and what is the reaction center of PSO? As of now, we are writing it down, so we will remember. Pisoleo napahoriba. If you have to be first, you always have to get more marks. Yes? Understood or not? I don't know. Right? So, P700, P660 is the reaction center. Second point likha photophosphorylation. Remember this word photophosphorylation? Photo is light, phosphorylation ki asile? Addition of phosphorus to ADP for the formation of ATP. So, we know since we are talking about light reaction or product ki ATP or NADPH2. So, ATP form koribo lagibo, kune ATP form koribo, ADP form koribo. So, in ADP phosphate needs to get added. So, what is the process that is going on is phosphorylation. So, we have phosphorylation taking place in presence of light. So, it is called as photophosphorylation. I think you remember we have talked about something called as oxidative phosphorylation also. Just to make a comparison, no? Oxygen or in presence of oxygen ATP যদি form করিছে তাক আমি কি বলি কম oxidative phosphorylation. In presence of light যদি form করিছে it is called as photophosphorylation. Oxidative phosphorylation something related with respiration and photophosphorylation related with photosynthesis. Clear? Etia as of now ekhini likhi lua, after that little later we will explain. আৰু এটা পইণ্ট আছে both stroma thylakoid and grana thylakoid আৰু হিপিনে present only in grana thylakoid. I think I can rub the first point, no? আৰু এটা point আছে, point number 5. We'll first write it down and then we'll explain. हाँ cyclic pathway লিখিছো দেই ব্র্যাকেটত So, ইয়াতে বহুত ইনফৰমেশন সোমাই আছে লিখি লোৱা উইল আন্ডাৰস্ট্যান্ড The fifth point is source of electrons. Source of electrons for PS1, it has two sources. Number 1 from chlorophyll A, bracketot likhe is a cyclic pathway. Number 2 from PS2, that is the non-cyclic pathway. And if you are talking about PS2, it has only one source from where the electrons come, and it is from photolysis of water. As we go about we'll be understanding PS1 separately and PS2 separately. First, আমি চাৰ্টখন লিখি লৈছো, পিছত আমি বুজি পাম. If it's over, please tell. Over, na? তোমালোকৰ হ'ল? Okay. What was the first point about the reaction center? I think the word reaction center is clear. In the group, the most important member is obviously chlorophyll A, and that is what is called as a reaction center. Reaction center naam ki diso P700 and P680. It could also be called as clear? P700 or P680 question, no? P700 could also be denoted as chlorophyll A700. Eketai bostu group to bitot sobotse important ki chlorophyll A. So chlorophyll A700 aru PS2 into reaction center that was called as P680. That is also called as chlorophyll A680. That's the first point. Clear? Okay. Coming to the next point, something about phosphorylation. Why we are talking about phosphorylation? Because we know during light reaction ATP gets synthesized. You have to know ATP code form korise, PS1 form korise na PS2. Who is associated? Which group of pigments is associated for the formation of the ATP? Both the groups are associated for the formation of ATP. So light reaction has been divided into two steps, cyclic or non-cyclic, or PS1 and PS2. Duita cycle ote, either it's cyclic or it's non-cyclic. Duita pathway te ki hobo ATP synthesize hobo. So cyclic pathway to ATP synthesize hobo, non-cyclic pathway to ATP synthesize hobo. So photophosphorylation be understood. Talking about location, since PS1 and PS2 are nothing but pigments, so pigments bilak kot pam. Ityalake ki koi asilo thylakoid, but again we have something called as a stroma thylakoid and the grana thylakoid. What is a stroma thylakoid and a grana thylakoid? If suppose we are talking about this as a chloroplast, individual jun bilak thake tak ami ki boli kom thylakoid, jun bilak thylakoid stacks out thake tak ami ki boli kom grana. Remember thylakoid and grana, no? So PS1 code thakibo, remember this word both. Etor code question I protect to point about this chart is something that comes in the exam, right? MCQ point of view for question size. So be careful with the words, right? So both stroma thylakoid and grana. So these are called as individual bilakok ki boli kom stroma thylakoid, grana jun bilak thake tak ami ki boli kom grana thylakoid. So yatu thakibo PS1, e bilakot thakibo PS1. But if you are comparing it with PS2, PS2 kot thakibo only in, right? Again the important word is, right? So tahate ei individual jimanbur ase ei individual bilakot PS2 na thake. PS2 kot thakibo jeti grana form korise PS2 okol grana the thakibo, kintu PS1 kot thakibo hey group 2 kot thakibo duita the thakibo, right? So remember the word both and only. Coming to oxygen. Why we have to talk about oxygen? Because we know photosynthesis is an oxygenic process. Apart from bacterial photosynthesis, baki photosynthesis ki hoy oxygen evolve hoy. So oxygen se evolve hoy. At one point of view, we have said that photosynthesis represents by two reaction, light and dark. You have to be precise. Kuntu reaction or oxygen evolve hoy, light reaction and dark reaction. So obviously light reaction, right? And I think you all know we have said something called as photolysis of water also. Light reaction on water molecule to bhangi jay and ultimately ki produce kore oxygen. So oxygen is a part of the photosynthesis reaction, light reaction form hobo. Oxygen evolve kundot hobo, PS1 is responsible and PS2 is responsible. It is PS1 that is responsible, right? PS2 oxygen evolution lokot eku link nai. We'll be talking about that process, right? Sorry, PS1 not evolved and PS2 evolved. Actually PS1 PS2 boli kwat ke the precise, I mean the best way of saying is all of pisot ami jintu discuss korim no cyclic or non cyclic. So question to ki ahe is the cyclic ot oxygen produce hoy na non cyclic ot oxygen produce hoy hetu bostu ami porhim. As of now oxygen evolution logot kun associated hoy ase PS2, right? And ultimately we come to the fifth point, that is the source of electrons. Source of electrons ER kothao in the first class common introduction we have done. So what we said is that we have a pigment group, right? Out of this group, so what's the important key? Reaction center. Reaction center could be P700, it could be P680. Light poribo, লাইট পৰিব লাইট ইয়ালে আহিব লাইট আহাৰ পিছত কি হ'ব ইলেক্ট্ৰন ওলাই যাব ৰাইট ইলেক্ট্ৰনটো যে ওলাই গ'ল দুটা পাথৱে আছে non-cyclic either it will come back to its original place or it will go somewhere else if it comes back that we call as cyclic or jodi belegole jai it is called as non-cyclic etia come to this point this is a molecule right chlorophyll a hoy he jodi electron to ulai die tar eta vacancy hoy gol ne nai positive hoy gol ne nai right kono to enekwa charged particle hoy thakibo ni bisare everyone wants to remain in a grounded state so what it does is since the electron has been ejected out it became positive so it is in search of an electron again to make it in a grounded state so he ki koribo kabar pora electron ta accept koribo lagibo tetia he ako normal ba stable condition ot thakibo light pora loge loge unstable ho electron ulai dile he electron eta accept koribo lagibo bahiror pora tetia he stable hobo so this is what we are talking about it as source of electron right so if you are talking about PS1 boli nokoi pelai actually what we are concerned with the reaction center so reaction center of PS1 e electron duta jagar pora pai either it gets it from that original cyclic pathway or sometimes it gets from the non cyclic also keneke pai as we go on drawing the diagram ami clear hom as of now just remember the reaction center in case of PS1 takes the electron or could take the electron from two sources and if you are talking about PS2 it will have only one source of electron right so hey statement to aru keneke clear hobo if you are talking about P700 no P700 ki hoy PS1 e ki release korile electron if it is a cyclic process a electron to yale ghuri ahibo nahe ahibo so etu jodi enekwa hoy it will come back mane etu nahe ultimately ahi ghuri ahi ki hobo jor pra ulaisil tale ahi jabo so ultimately it has got it from its own so we have written that PS1 or reaction center electron to core pra pai from chlorophyll A itself jor pra ulaisil tale gusi ahe right that is one source or as we go about in the lesson ami pisot gom pam say a electron to ketiaba P700 le nogoi polai P680 leu gusi jai if you are talking about non cyclic jedi cyclic hoy he ghuri ahibo jedi non cyclic hoy he belegole jabogoi right so it will have this one as actually as of now it will be better you write it down when we draw the diagram it will become more clear right etia khali etai monot rakhiba jedi electron ulaise tetale electron accept koribo lagibo PS1 e electron accept duta jagar pora kore PS2 e electron accept eta jagar pora kore clear keneke kore ami diagram akim so this was the comparison those who have read this chapter non-cyclic photophosphorylation বা non-cyclic pathway to jedi monot ase non-cyclic ot ki hoisile electrons ulaisile from PS2 right okay forget we'll write it down and then we'll explain again it has some more points about PS1 and PS2 individually chart kon overhaul eta PS1 boli heading koribason aru keta man extra point likhibo laibo What is PS1? It is a group. So, the group will contain different types of pigments, not only chlorophyll A, but also some other pigments. So, PS1 is a group of pigments, PS2 is also a group of pigments, PS1 or ki thakibo essential pigment, accessory pigments, together with that iron sulfur proteins thakibo, FES protein, and later we will see that FES protein is considered as the first acceptor of electron. Etu pisot buji pam. Comparing with PS2, PS2 also contain a single molecule that is called as a reaction center. It is denoted by chlorophyll A680. It also contains chlorophyll B and carotenoids. Together with that, it has other components also, Mn2 positive, chlorine, quinone, pheophytin, etc., right. Now, from this part, questions ki ahise? First, reaction center to upor ahise, PS1 reaction center ki, PS2 reaction center ki. Second question has been asked about the amount of chlorophyll B. Amount of chlorophyll B code basic ase, PS1 or basic ase na PS2 basic ase, right. So, please read all the informations very carefully, right. So, sometimes the question has been asked about the number of chlorophyll B molecules also apart from chlorophyll A, right. And similarly, we have all this, and since we are talking about water oxidizing enzymes, so water oxidizing, tamne water to bhangi diya enzymes kisuman, and since what we are referring to is nothing but as photolysis. So, photolysis or karone, who is responsible? PS2, clear? So, pigments thake, pigments বিলাক groups ot thake, keita group, duta group, what are the names of the group? PS1 and PS2, right? PS1 have a important member, PS2 also has an important member. That is what is called as a reaction center, and the other components is something that we have discussed. Etia ame ki korim je photosynthesis process to keneke hoy. We have to talk about the mechanism of photosynthesis. First we will be discussing the light reaction and then we will go for the dark reaction. So, heading koriba mechanism of photosynthesis. Is this over? Online students over? Musiye dilu atha. Okay. তোমালোকৰ হ'ল নাই হোৱা হ'ল নেকি তোমাৰ হৈছে Just a second. Light reaction, also called as Hill reaction or generation of assimilatory power or photophase. Yeah. Arnold or Emerson ki kosile light reaction occurs in 2 steps. Which 2 steps we are talking about? Cyclic and non-cyclic, right? color nahi. Can you see the diagram clearly? No, it's confusing. Pele kya ki diyo niki okman? Dangor kya ki diyo niki? Okay, let's not draw the diagram. Okay, okay. Likha ki nahi hoise ne? Okay, likhi lua. So these few lines talks about the light reaction. Light reaction ketia hoy as soon as the light falls, right? What happens during light reaction? During light reaction, transfer of light takes place, right? So light reaction ki hoy? Light transfer hoy. Kar pra kole? Obviously sunor pra kole pigments hole. So that's the diagram we are going to draw. लिखा होल निकी? Okay. So what happens during light reaction? Transfer of light energy takes place, and transfer to, it could be denoted in a diagram. हाँ So, a diagrammatic way to represent a light reaction, what happens or when does light reaction begins? The light reaction begins as soon as the light falls. Who will take in the light? The pigments will take in the light. Pigments keneke arrange hoye in the form of photosystem, right. So, what we have drawn is a photosystem that comprise of the accessory pigments and the essential pigment. As soon as the light falls, actually light reaction of ki hoye ase? Transfer of light energy. Transfer of light energy and converting itself into electrical energy hoise nai. Electrons are coming out, right. So, light energy ki hole convert hoise? Electric energy le. Yate hoise ki transfer. When we are talking about transfer of light energy, no, two types of transfer could be observed. One is called as the resonance transfer and the other is called as the inductive transfer. Resonance transfer ki aru inductive transfer ki. Transfer within the accessory molecules, tak ami ki boli kom? Resonance, gutei light khini prothomote tahate loi pelai, ultimately tahate ki kore? Chlorophyll 1 di diye. Aru chlorophyll 1 di diye process to kami ki koise? Inductive resonance or inductive transfer. Why doesn't chlorophyll A take the light directly? Photooxidation, right? Direct light to loi lole ki hobo? Break hoi jabo. E gutei pigments bilakor bhitor sobotse important etu nohoy jano. So, here jodi bhangi jay photosynthesis keneke hobo, right. So, ultimately this pigments what they will do? Protective pigments, accessory pigments are also called as protective pigments. So, they will protect this against what? Against high light intensity, right, and the light will get transferred. Two types of transfer. Accessory pigments to reaction center, hetu kami ki boli ko? Inductive transfer. Clear? Okay, you have something called as LEC. A word to janibo লাগিব, light harvesting complex, right? E gutei khini ami ki boli koisu? Light harvesting complex boli koisu. Is the diagram over? Also? Is the process understood? Okay, diagram to tolot ako points continue koriba. Is it clear? So, ultimately electron to cyclic and non-cyclic pathway jabo. Okay. Next heading kora cyclic and non-cyclic photophosphorylation. Whole last point too. চার্ট এখন লিখা। Right. So, I think you are just getting the idea, right. So, light porile, light porar pisot ki hol electrons eject hol, electrons kot jabo cyclic aru non cyclic. During the process ATP gets synthesized, and so we are writing the word cyclic photophosphorylation and non cyclic. So, we will be comparing the two processes. So, products synthesized during cyclic photophosphorylation only ATP, in that case both. Next, oxygen evolution is related with cyclic and non-cyclic, it is related with non-cyclic. Photosystem required, cyclic process requires only one photosystem PS1, it requires both the photosystem PS1 and PS2 because the electrons will move from one photosystem to the other photosystem. As far as source of electrons are concerned, since we are talking about cyclic, it will come or it will come to the original place. So, belegor pra tahate electrons to bole dokar nai. But since we are talking about non-cyclic, the electron will not return to the point of origin. So, it requires an external source. Who will give the electron? It is the water by the process of photolysis. If you remember, we have talked about photolysis, okoman ke koisilu. Water molecule gets broken down into electrons, protons and oxygen. Oxygen is a gas, so it will move out. What will happen to the electrons? It will be accepted by the PS2. Right? When we draw the diagram, it will become more clear. Ekhini loke kiba doubts ase neki koa? Eta sequence of photosynthesis process to buzi paisa ne nai? I think it's almost, the time is over. So, we will wind up the class here. In the next class, আমি কি discuss করিম cyclic or non-cyclic in details with a proper diagram so that we understand, right? কেনেকে electron to move করে, কোলে যায়, কোনে accept করে, right? আর finally কোলে যায় গৈ, final electron acceptor, primary electron acceptor, etc., etc., right? So, we will be drawing the sequence. Up till here, কিবা doubts আছে যদি please ask, or else we can just wind up the class. I think it's clear. Right? Thank you.